All right, so hope you liked that video. That's one of my favorite videos. Um, just an awesome rap about cellular respiration. I know a lot of that you might not know, but actually, after this unit, you will recognize most of the things that they've said in that rap. And maybe, if you're lucky or not, you may be making your own songs, poems, stories, or creative analysis of parts of cellular respiration. So, we have already talked about energy. We talked about reactions that give off energy. We talked about reactions that absorb energy from their surroundings. We've talked a lot about cell membranes and how things can move through cell membranes. We've talked about the movement or diffusion of water across membranes. We've talked about macromolecules and parts of the cell. And we talked a little bit about chemistry. And one of my favorite things about this unit is it's going to take everything that we've learned and it really combines it into a way where you can see how all of these parts of biology that have been important to us are going to manifest themselves into living, working cells. Cellular respiration is a process that happens in both in the cytosol and the mitochondria. It's a process of catabolism. It's a process of breakdown whereby the starting molecule is the glucose molecule, which you know tons about. Um, if you want some extra review, uh, you can look through chapter 6 in your book. It should have some help. It's not going to go into the same depth that we will go in during this unit, but it might help you put things together. And if anything, it's going to help you for an overview. But we know all about energy. We know all about the fact we know that molecules have free energy, and they have resonant energy. That energy that they have as a result of the bonds or types of atoms or even the arrangement of those atoms is very important to a molecule's resonant energy. And cells are going to use this energy to harness more energy from in a way that they can use. So this is all chemical energy that we're talking about. Energy that exists in the bonds, arrangement, and atoms of organic molecules. Cellular respiration has three major parts: glycolysis, the Krebs cycle, and electron transport chain. Glycolysis occurring in the cytosol of the cell, where it's the breakdown of glucose to 2-pyruvic acid molecules, whereby carbon dioxide is then given off in between the Krebs and glycolysis. We make some energy. We've got more breakdown of energy here. And ultimately, all of the energy that we're harnessing from the molecules of glycolysis and Krebs cycle will generally go to this place called the electron transport chain, which is also called oxidative phosphorylation. We talked a little bit about phosphorylation. It's just putting phosphates on or off molecules. And you're going to see some clear examples of phosphorylation and dephosphorylation in this coming unit. So it's all about energy. It's all about energy. Even plant cells have mitochondria that break down glucose that is made by the chloroplasts. So both plant and animal cells carry out this process. Whereas the animal cells they take in oxygen, release carbon dioxide. The carbon that comes out of the human body when you breathe, when you exhale, is all carbon that's been used and abused and broken down to its most basic form from sugar molecules. And this all happens in the cytosol and the mitochondria in the animal cells. So water and carbon dioxide are byproducts of that. On the other hand, plants use, all right, they use carbon dioxide and water and sunlight's energy to make glucose, but they also use glucose in the form of mitochondria cellular respiration breakdown. So whereas the process here is called photosynthesis, the process in blue is called cellular respiration, and it's basically recycled from animals and plants. So the air that you breathe in and out is being taken in and out by plants, and the plants, the things that their waste products are the starting material for some of the things that mammals and organisms use. to break down energy. So it's always one, always one. If you think you're separate from plants, you're just not. We're all breathing the same stuff, and we're all recycling the same products. One of the things that's important is that cellular respiration requires oxygen, the presence of oxygen. It's an oxidative mechanism, meaning that it's going to accept electrons from other molecules. All right, you know oxidation and reduction. All right, so oxygen becomes reduced, and oxygen ends up oxidizing some other molecule. If there is no oxygen present, a process called fermentation will occur, and we'll talk about specifically what happens when organisms, especially mammals, don't get enough oxygen in order for this process, cellular respiration, to occur. Cellular respiration is a high energy output process, whereas fermentation is a lower energy process. Fermentation might be the more primitive of the two, though, because most organisms have some fermentation mechanism, especially prokaryotic organisms, and some eukaryotic organisms can carry out both. But cellular respiration definitely yields the most energy of the two processes. So where is the energy in this glucose molecule here? A good question to ask yourself, just for some review, is whether or not this molecule is a beta or alpha glucose. All right, take a look at it. Look at the number 1 carbon. The hydroxyl is in the up orientation, so we know that this is a beta glucose molecule. Where is the energy here? The energy is in the bonds and arrangement of these organic molecules. I think I stated before that alpha and beta glucose, although they're hydroxyls and hydrogen on the number 1 carbon in different places, they're structurally very similar, but they're not going to be on the same energy level. Now, I don't actually know which one has more or less resonant free energy. But it just goes to show that the arrangement of atoms and molecules can lead to different atoms and molecules having more or less energy states. Molecules that have more energy to give off and be used, and molecules that have less energy have less energy to be used by the cells. So the most important thing about cells with this molecule is it can be strategically and stepwise broken down slowly, whereby the electrons are going to be taken from the hydrogens of this molecule to be broken down. All the oxygen and the carbon will then be released as carbon dioxide, especially in animal cells. All right. Basically, the idea of cellular respiration is about the fall of electrons. Electrons, we know electrons have different energy states, all right, because electrons occur in different orbitals in atoms, and each orbital has electrons of a different energy state. The farther away an electron is from the nucleus of the atom, notice I said, the more energy it has, meaning the more energy it has to be able to stay a part of the electron valence spectrum. So it's all about electrons. Just remember that. Although cellular respiration is a highly complex process, it's not confusing. It's a process that occurs in steps. And one important thing to remember is that this process is all about our electrons. All right, it's all about our electrons. It's all about using electrons from the energy that they have, and every step here we're going to harness and take away energy from the electrons to make energy in the form of ATP in some way. We know adenosine triphosphate. It's the energy currency of the cell. All right, and then this ATP will end up doing most of the cellular work, although electrons themselves will do a good amount of work. The entropy given off is going to be used to create heat, which can also be used to keep organisms at a high temperature, which is what they need. All right. Oxidation-reduction reactions drive cellular respiration, and I said it's all about electrons. So basically what we're going to be doing is taking electrons from certain molecules and putting them onto other molecules. All right. Now, in order for electrons to jump from one molecule to another, one molecule has to have a greater pull for that electron. All right, it's called electronegativity. All right. So what we're going to be doing, we're going to be carrying electrons on these things called electron carriers. All right. And we're going to be allowing those electrons to move to other molecules so that they can be moved to different places. All right. Now, when molecules put electrons on other molecules, all right, they're called the reducing agents, and the molecules that actually got reduced, all right, or are taking electrons from the reducing agent are called oxidizing agents. All right, this is difficult. Just know that the molecules, cells, or things that accept electrons are reduced, and the things that give off their electrons and had them but no longer do are oxidized. All right. So, like I said, it's all about using electrons. Well, we have to find ways to move electrons from one place to another, especially because these electrons are going to be falling in energy states. The electron here is high energy, the electron here is low energy, and we can't treat them the same. All right? We have to treat them differently based on their energy states. All right. So we got oxidation reduction, electrons moving from one molecule to another. It allows us to use and move energy in a more sophisticated, that's a horrible word to use, sophisticated, efficient way. So we're going to be using electrons. So everything you see here is what we're really doing is finding ways to harness as many electrons as possible, as many electrons that are as high energy as possible, and using them for their energy states, and then once they don't have no more energy, we're going to kick them to the curb and get rid of them. All right, that's basically what happens. All right. One of the most important molecules that we're going to see is called NAD plus or NADH. I think it's called nicotinamide adenosine dinucleotide. All right, nicotinamide adenosine dinucleotide, nicotinamide adenosine dinucleotide hydrogen. All right. So there's two forms of NAD. NAD is important. It is our primary electron acceptor. All right. Say it's a primary electron acceptor. It is, well, I shouldn't have said that, sorry. It is our electron carrier. All right, it is our primary electron carrier. What it does is, in this form, oxidized, NAD+, it accepts two electrons, all right? 1, 2, these red dots here. And it accepts one hydrogen. So it accepts this hydrogen with its electron, and it also accepts an electron here, but it releases its hydrogen ion. So it releases hydrogen ions into the solution. So nicotinamide adenine dinucleotide hydrogen is the reduced form where it contains two electrons, and nicotinamide adenine dinucleotide without the hydrogen here is the oxidized form. All right. So what this molecule is going to be doing, it's going to be providing a place for electrons to be safe while also get carried from one place to another. You don't want electrons roaming the cell on their own. They're going to be reacting with things in the cell that are important. They're going to cause disruption. There are also these things called free radicals that you've probably heard, really important health-wise. You don't want ions roaming the cell on their own. So this molecule is specifically designed to carry electrons. All right. Now, how do we get those electrons from one molecule to another? It's all about electronegativity. All right. This chart is just showing the electronegativity that certain atoms have for electrons. All right. So silicon has sodium and lithium and silicon all have a low electronegativity, as opposed to chlorine, nitrogen, oxygen, and fluorine all have very high electronegativities. All right, so you can imagine that an electron can very easily move from lithium to beryllium, and it can very easily move from beryllium to nitrogen, and it can very easily move from nitrogen to oxygen and move to oxygen to fluorine. Electrons will not, on their own, be jumping from fluorine to nitrogen, or from fluorine to boron, or from nitrogen to lithium, or from carbon to beryllium, because all these atoms to the right have a greater electronegativity for electrons than those to the left, which means that an electron is naturally going to jump this way onto this one because this one has a greater pull, and then it's going to jump here, 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 here. So this is one way where we can actually allow electrons to move from one thing to another. So all these organic molecules that we're going to see might have some of these different elements that allow electrons to jump from one place to another. All right. Now the interesting thing is that an electron at a very high energy state, all right, is going to jump differently than an electron at a very low energy state. An electron at a very low energy state is going to need a more high electronegative atom to get that electron to jump to it than would be for lithium. All right. So electronegativity is important because it allows electrons to move from one molecule to another. But it's important to notice that jumping of electrons from one molecule to another is very, very important in cellular respiration. Electrons being carried from one place to another is generally done. The universal carrier is NADH in the reduced form. And electronegativity is very important. We talked about ATP. It's the chemical energy. It's the chemical energy currency of the cell. It's a great way to store chemical energy. Whereas NADH is used to carry electrons, it's not really used for its energy. ATP is used as a chemical energy storage molecule. All right? And the most important part about this molecule is the storage of energy in this bond here, from phosphate number 2 to phosphate number 3. Again, each phosphate you put on this molecule, the more unstable this molecule becomes, but the more energy that it has to release to be used for chemical reactions. This is all called reaction coupling. All right. There's two different types of phosphorylation. One is called the inorganic phosphate addition, and the other is called substrate level phosphorylation. I didn't show anything, but a substrate level phosphorylation is when a phosphate comes from one organic molecule and it gets put on another organic molecule. That's called substrate level phosphorylation. And another phosphorylation is called the inorganic phosphate addition, which is a phosphate functional group coming from solution, like it's roaming the cytosol, and then it gets put by an enzyme onto the ADP molecule to become ATP. All right. So this is our energy storage and uses molecule. So now we have molecules that are designed to provide energy for cellular work, like ATP, and then we have molecules that are designed to carry electrons from one place to another. Does anybody know which way south is in this picture? Which way is south? Which way is south? And how do you know? Interesting. Which way is south and how do you know? Oh, the questions. Okay, so let's review a little bit. Cellular respiration is a process using oxygen as an electron acceptor in order to create energy, right, in the form of ATP. Whereas fermentation is we want to make energy in the form of ATP, but we do not have oxygen. So think of holding your breath. Your cells still have to work, but they're not getting oxygen. They may still make some energy a little bit more inefficiently, but we can still do that. Right? Fermentation is the more primitive process. We have organelles called the mitochondria and the chloroplasts. Both have different functions, but very similar in kind of what they do and how they do things. We have carbon cycling between plants and animals, between carbon dioxide and oxygen. We have fuel or chemical energy. We looked at the glucose molecule and all the chemical energy that glucose molecule has. We just talked about the role of oxygen in cellular respiration. If we go back, oxygen is actually a very electronegative atom, which is why water is so polar. And is why many things often that have oxygen in them are electronegative or have polarity to them because oxygen is so electronegative. Loves to pull electrons. Now, electrons are basically what cellular respiration and fermentation is designed to use in order to get that chemical energy. The chemical energy is going to be in the form of electrons or the using of electrons. And interestingly enough, when you take an electron from its hydrogen, you end up with a hydrogen ion. All right. Which is basically hydrogen ions without their electrons, which are going to be used and are a very huge part of cellular respiration. We're going to talk about that. Talking about the fall of electrons from high energy to low energy and the cell's ability to use this fall of energy states in order to do work. Talked about transporting electrons a little bit using that molecule called NADH, nicotinamide adenosine dinucleotide hydrogen. We have storing chemical energy. When you talk about storing chemical energy and doing cellular work, you should be thinking ATP. If you're thinking fats, then thinking glycogen, then you're also thinking correctly as well. We have oxidation and reduction is going to be our primary means of taking electrons and allowing them to move from one molecule to another. ATP is hugely important. Most of its chemical energy that is going to be used by the cell is going to be stored in the bond between the second and third phosphate, the very negative region of the ATP molecule. Wow. Woo! Whew, are you ready for this? Man, that was a lot of stuff. But generally, in review, a lot of things we've talked about. We've talked a lot about membranes. We've talked a lot about macromolecules. We've talked a lot about parts of a cell. We talked a lot about ATP, reaction coupling, and enzymes. All of these things are going to be working in unison with one another to create the perfect, most efficient way of making energy for the cell. Because remember, living things are low entropy. Living things are ordered, and it takes lots of energy to keep things ordered. So the cell is going to have devised over hundreds of millions of years, even billions of years, the most efficient ways. And don't forget, this is all being done and helped out by lipids, proteins, sugars. Steroids are sending signals. Enzymes are doing their work. The DNA is sending messages. Things are reacting. They're signaling. There's intrinsic and extrinsic membrane proteins. There's exocytosis and endocytosis and hormone signals being sent from one place to another. This is insane. The functional groups matter. These things make organisms do what they do. So I apologize if this is a little bit long-winded, but this will be the end of part 1. And after this, we will be getting into part 2, which will probably, I haven't looked yet, be the stages of cellular respiration and hopping into a process called glycolysis. So thanks for listening. There should be some questions for you to answer as you go through this mini lecture. Hopefully it was mini enough, and I hope it wasn't too long. And yeah, I'll see you tomorrow. Alright, isn't that enough fun for one day? If you haven't heard that song, you know it now. Awesome song. Thinking about trying to rework that song to talk about cellular respiration, but we'll see. We'll see if I have the extra time. But so this is the beginning, officially the beginning of part 2, which is again about cellular respiration, but it will be focusing on this process here called glycolysis, glycogen, glucose, lysis to break, which we're gonna just break glucose into a bunch of pieces to make some energy. But generally, what we're doing here, we're gonna create a few ATP. We're gonna create some molecules that we need. Now, in very simple organisms, glycolysis is the only process that some organisms have, like some bacteria. Organisms that don't have mitochondria only perform glycolysis. It's a process called fermentation when only glycolysis is used. Generally it occurs without oxygen, but it can occur in the presence of oxygen as well. And the first one we're going to focus on, it's the first of 3 major parts for cellular respiration. A couple things to remember. Cellular respiration is all about harnessing chemical energy in the form of ATP from things like electrons in the presence of oxygen. Glycolysis is our first one. Let's kind of hop right into it. These are all the major cellular pathways. I think we saw this before. This is in the center here, this is the backbone of metabolic pathways. Pretty much every other pathway is in some way connected to this process of glycolysis. You're going to see glycolysis. This stuff in here happens in the mitochondria. ATP synthase. This is called oxidative phosphorylation, also known as electron transport. Have a bunch of other different things going on, but it's just the different ways that cells use the energy to store things, for anabolic ways, for catabolic ways, and overall metabolism. So let's talk about glycolysis first. Again, glycolysis is the process of breaking down glucose in a series of steps. When you light a fire, what you're generally doing is starting a big fire that has lots of entropy, which means energy is being released into this atmosphere. It's a very exergonic reaction, but if we were to actually harness the heat from a fire for other chemical reactions, we'd be able to fire a lot of different chemical reactions from this major exergonic process of breaking down glucose to CO2 and water. But what cells do, cells break the combustion process of glucose into many different steps. In glycolysis, there's about 10 steps, 10 different ways in which the cell is going to set up molecules that are going to be used for energy later on. So the process of glycolysis takes glucose, brings it into the cytosol of the cell, breaks it down in a bunch of steps, releasing a little bit of energy along the way, but basically setting up another organic molecule called pyruvate to then either in the presence of oxygen be pulled towards the mitochondria to get broken down into more energy, or in the absence of oxygen will take a place of a process called fermentation, which is lactic acid or alcoholic fermentation. So let's focus on glycolysis. Glucose is then going to enter the cell by either diffusion through the cell membrane, diffusion through GLUT proteins, facilitated diffusion, still passive, or it can be actively taken into the cell if the cell really wants it. But generally, glucose will diffuse into the cell from the bloodstream by a process of the GLUT proteins. And so glycolysis is going to be occurring in the cytosol. It's a very primitive group of reactions, meaning these reactions have been taking place since the first very simple cells, the prokaryotic cells, have existed on Earth for a few billion years. You start with one 6-carbon glucose molecule, and what you're going to end up with is two 3-carbon pyruvate molecules. You're going to see what this is in a second as products. ATP is both going to be put in and taken out from this process, and we're also going to result in some NADH molecules being there actually are no inputs. I shouldn't have put that in there. There's only NADH outputs in this process, which means we're going to gain NADH. And if you're thinking electrons right now, then you're thinking correct. All right. So we take ATP, which can be directly used for cellular work. It's not that much though for this process of glycolysis. But we're going to circle here a bunch of times. NADH is one of our major electron carriers. It's going to be taking electrons in the form of hydrogens off the glucose molecule to be used for chemical energy. Isn't this crazy? If you can just think for a moment, just one moment, that right now your cells are carrying out these crazy processes and that all it is made of carbon, hydrogen, oxygen, nitrogen, all these different other atoms that work together to do this crazy, I want to say a swear, but I can't. Especially not unrecorded. So it's just crazy how this happens. Just, oh, it's so crazy. This process, it's insane how it works. So here's the process of glycolysis here. There you go. We start off with our glucose molecule, alpha or beta. Yep, we start off with an alpha glucose molecule. And you'll see in a series of steps, we're going to change the molecule into different things, which will generally end up as two 3-carbon molecules. So here's a huge thing to understand. We start with a 6-carbon molecule, 6-carbon molecule, 6-carbon molecule, 6-carbon molecule. Then the 6-carbon molecule gets split into 2. So now we have two 3-carbon molecules, DHAP, dihydroxyacetone phosphate, and glyceraldehyde 3-phosphate. All right. But dihydroxyacetone phosphate actually gets taken in one more step, gets changed over to glyceraldehyde 3-phosphate. So after step 5, everything that happens here happens twice. Okay, that's very important. Very important. This process of glycolysis we have 6 carbon, 6 carbon, 6 carbon, 6 carbon, 2 3-carbon molecules. This 3-carbon molecule gets changed into this one by an isomerase. It makes an isomer. An isomer is a molecule that has the same chemical formula but different structure. That's what this one is. If you count everything up, it matches up to this one. So this one's going to be changed into that one. Now you have 2 of these. Now everything that happens will happen twice. That's important. All right. Another thing to understand is this is all drawn by cellular needs. All right. So basically, these reactions, all the arrows don't show, these reactions can actually go the other direction if needed, especially for plants who actually are going to be building glucose molecules, and especially if we want to be building things like glycogen from other products. But I know it's difficult. You're going to get overwhelmed right now, but the important thing is to understand what's going on step by step. Okay, if you can do that step by step, don't try and understand the whole without understanding its parts. All right, the whole is easy. The whole is the easy part. It makes electron carriers and a little bit of ATP, and ends up breaking down glucose into 2 pyruvate molecules. All right, but let's get kind of to the nitty gritty here. Right, we start off with our glucose molecule. All right, some important things are that glucose is a very high energy molecule. The first thing that happens here is the cell is going to take an ATP, a high energy molecule, and it's going to slap a phosphate onto the 6 carbon. All right, so that's the first thing that happens. This is called a substrate level phosphorylation. All right, because not because it's an even though an enzyme is involved. All right. But it's called a substrate level phosphorylation because a phosphate is coming off of one organic molecule and being slapped on another. And this is done by an enzyme called hexokinase. Hexo means 6. Kinase, all right, means putting on or taking off phosphates. All right. So when you see a kinase, all right, these are enzymes right here, up here, these names. Up here, these are enzymes. Down here is the name of each molecule. All right. So what you're looking at here is hexokinase basically takes a phosphorus and puts it on a 6-carbon sugar. All right, hexokinase, 6-carbon phosphate. All right, puts a phosphate group, OPO32- onto a glucose molecule, and now becomes something called glucose 6-phosphate. One thing to understand too is that this is a exergonic reaction. It means the second glucose enters the cell, this will almost immediately happen. All right, it will happen spontaneously. The reason why this is important is because if glucose goes into the cell, it might also try to diffuse out of the cell if the conditions are right. So by putting a phosphate group on this glucose, you actually prevent the phosphate from leaving the cell, or the glucose 6-phosphate from leaving the cell, because this phosphate is highly charged and it's not going to go easily through those glucoproteins. It's definitely not going to go through the membrane. All right, we know that now, hopefully. So all this stuff matters. All this comes together. All right, and it's also important to know that the first 5 steps of this are called the energy investment phase. All right, we're investing energy. We're going to put energy into the glucose molecule so that at some point we can get as much energy out of the glucose molecule as possible. Okay, so first step, hexokinase catalyzes a reaction where a phosphate is taken off an ATP, put on glucose to become glucose 6-phosphate, and it becomes ADP, adenosine diphosphate. Now this reaction is exergonic, all right, on its own, but also with the help of ATP becomes even more exergonic. Now, if we go to our next step here, the first 5 are the energy investment phase. Now we have glucose 6-phosphate. All we're going to do here is we are going to make glucose its isomer called fructose. All right. The only difference between glucose and fructose is that glucose has 1, 2, 3, 4, 5 carbons in the ring. Fructose only has 1, 2, 3, 4 carbons in the ring, and it's got 2 carbons outside the ring. All right. Now, why do you think this is going to be important? Well, If you said that the molecule looks much more symmetrical, then you'd be right. I know you probably weren't thinking that. All right. It's important because we're making this molecule look more symmetrical, because what do you think is going to happen in a couple steps? Well, it's going to be broken into two. Well, if you make the molecule more symmetrical, when you split it, you're more likely to have two equal parts. So we don't want two totally different molecules, because then we'll have to spend energy on doing two completely separate reactions, all right, to get energy from them. So the point of this is to make glucose a high energy molecule by putting another phosphate on it. We change its form, all right, it's called an isomer, all right. So glucose and fructose both have the same chemical formula but different structures. So it's done by an enzyme called an isomerase, all right. So isomerases are enzymes that make isomers. Basically, they change the structure of one molecule and they put it into the structure of another. And it's called phosphoglucose isomerase because it's taking a glucose with a phosphate and it's making its isomer, fructose 6-phosphate. All right. So now we have a molecule called fructose 6-phosphate where we have phosphate on the number 6 carbon still, and we have a carbon that goes from inside the ring to outside the ring. All right. Now, the next reaction, all right, is the process of putting another phosphate on there. Now, if you're thinking, why are we going to do that? All right. All right, I apologize about that. I was talking about fructose 1,6-bisphosphate. Just got a call from my brother. He lives in Tacoma, Washington, so I don't see him too often. So I had to take that. But, you know, got to put that energy into your relationships. That's right. So speaking of energy, fructose 1,6-bisphosphate. You can see one of the only reasons that we've changed glucose to fructose is so we get a more symmetrical molecule by which we can put two phosphate groups onto its carbons, and when we split it, we'll actually end up with two very similar molecules. Now you can see really the only difference between these two molecules, there's a phosphate group on one of the carbons over here, there's a carbonyl on another over here, and there's a hydrogen hydroxyl carbon, which is both of these. So they're actually not very different, but this is the molecule glyceraldehyde 3-phosphate that we want. So far what have we done? We've rearranged glucose a little bit. We've used 2 ATP. So right now we're negative 2 ATP. We haven't made any energy. But like I said, this is the energy investment phase. All right. So we're investing energy. That's what we're doing. So we're going to get some back later on, hopefully. There's another little step here. Again, we have an isomerase, which is going to take one molecule and change it into its isomer. So they are the same chemical formula, different structure. So now we have 2 glyceraldehyde 3-phosphates. All right. This next reaction is one of the most important reactions of glycolysis. It's where we start to reap the benefits of what we've done. So we start out with glucose, but now we have a very high energy molecule. Yep, gotta love those motorcycles. We have a very high energy molecule here with 2 phosphate groups, much more unstable. It's going to split very easily with the presence of the aldolase, even though it still is an endergonic reaction. We've now changed it into glyceraldehyde 3-phosphate, which has the phosphate groups. This next reaction is called glyceraldehyde phosphate dehydrogenase. All right, whenever you see a dehydrogenase, all right, you're going to see hydrogens being taken off molecules and put onto something called NAD+. And now I think you know what NAD+ means. This is a very important electron carrier. So now we've taken a hydrogen, all right, and its electron, and actually we're going to put that hydrogen and its electron onto the NAD+ molecule. So now we're carrying electrons as well. Now, if you notice here, we have a pi going into this reaction. In this reaction, what's cool about this one is we don't actually have to use an ATP to get this phosphate on. All right. But the crazy thing, you might be wondering, why are we putting phosphates from ATPs on these? All right, we're using ATPs energy to do that. Well, here we're only using an inorganic phosphate. Why can't the cell just do that? Well, these molecules here are way more high energy compared by themselves than the glucose is up here. So the important thing is that the resonant energy, the free energy of these molecules, results in the ability to just take some random phosphate functional group and slap it onto the number 1 carbon here. So now we have a molecule called 1,3-bisphosphoglycerate. All right, so it's got a phosphate on the 1 and the 3 carbons, basically it's a much more high energy molecule now. And remember, all this from 6 on, everything's happening twice. All right, so now we've gotten some energy from the electrons. We have a molecule with 2 phosphates on it, very high energy molecules, and the next reaction includes something called phosphoglycerate kinase. All right. Now you probably know what a kinase is. Kinase involves a phosphorylation or a dephosphorylation. And what's going on here is a phosphate group from the number 1 carbon. This phosphate group is getting put onto ADP to become ATP in the presence of an enzyme called phosphoglycerate kinase. So now we've netted. Well, now we're yielding right in this reaction specifically. We've yielded 2 ATP because remember, everything's happening twice. So even taking in 1 phosphorylation in step number 7, we have now equaled the amount of energy that we put in. So now our net energy is 0. Right? We're 0 net ATP, and we're +2 net NADH plus H pluses. Okay. Now what we're going to do in these next 2 reactions, we're going to switch. We're going to take this molecule 3-phosphoglycerate. All right. We're going to mutate it, meaning we're going to change a couple groups, the 2 and the 3 carbon switch. All right. And the next reaction is going to remove a water molecule. All right. So this water molecule is a waste product. All right. It's the first waste product that we have. And the next reaction, now we have something called phosphoenol pyruvate. All right. So we have carbon double bond to oxygen with an oxygen. We have carbon bond to an oxygen and a phosphate and a CH2 group. It's called phosphoenol pyruvate. Now the next step, another kinase enzyme called pyruvate kinase, it is going to take a phosphate off the number 2 phosphate off the 2 phosphate, and it's going to put it onto another ADP to make ATP, and this happens twice. So we net now, after glycolysis is over, 2 total ATP. All right, so we've netted 2 ATP, we've used 2 ATP, and we've made 4. All right, so we're netting 2 ATP and 2 NADH. All right, I know this is very difficult. All right, one important thing about a kinase enzyme is the kinase enzyme is always named after the molecule at which you end up with after the reaction. So this is called pyruvate kinase, not phosphoenol pyruvate kinase. Here you have phosphoglycerate kinase. They're called the same thing here. So phosphoglycerate kinase. Where's the other one? Other one's very similar. Phosphofructokinase. Phosphofructokinase. All right. So a couple things that you should start building to understand are, number 1, what is the point of each reaction? All right. Make this one high energy. Make it more symmetrical. Make it high energy and symmetrical. Split it into 2. All right. Yield the electrons. Take phosphates off to make ATP. Change it around a little bit. Change it around a little bit. Take an ATP off. All right. Now we end up with a molecule called pyruvate. And if you notice, there's not that many hydrogens on this. All right. So this is a very, very oxidized molecule. All right. We've taken a lot of energy out of it. But this molecule is going to be very, very important in harnessing chemical energy from both this molecule and electrons that are going to come from it later. All right. So if I would focus on number 1, why does each reaction happen? Number 2, how are the enzymes important specifically? Number 3, how much ATP is used? How much ATP is made? Number 4, how many electron carrier molecules do we create? How many do we use? More important things. Let's see kind of what's important. Where does all this happen? Don't forget where this happens. It happens in the cytosol. Don't forget that this part of cellular respiration, glycolysis, isn't really designed to net that much energy. For some organisms can live off 2 ATP per glucose, while most multicellular organisms cannot. So think about that as well. And let's see where we are. Okay, some things we should think about. Where is it happening? Think about the enzymes and why they are named the way they are. The enzymes tell you a lot about the reactions. we split glucose into two. What are phosphorylations and dephosphorylations? What's the difference between a substrate level phosphorylation here, all right, versus a inorganic phosphate addition in step 6, all right. The energy investment phase, all right, 1, 2, 3, 4, 5, versus the energy payoff phase, 6, 7, 8, 9, and 10, all right. Why do we phosphorylate glucose? What are some things that help? Why make things into fructose? Why is pyruvate our ending molecule? All this stuff is very important and kind of rounds us out. And before we actually get into the next phase, I'm sitting in the morning facing the eastern sky. The darkness fades to light and I'm feeling that energy. The rising sun's reactions lead to entropy. The nature of the universe is entropy. Enthalpy, temp, and entropy too. Delta G. Molecules favor disorder, it's their melody. Human cells control things using ATP. The nature of the universe is entropy. Enthalpy, temp, and entropy lead to delta G. All right. So now we're getting to some reactions called post-glycolysis reactions and pre TCA. These reactions happen in between the end of glycolysis, where you have two pyruvate molecules, 2,3-carbon pyruvate molecules between here, and before you really get into the citric acid cycle. The pyruvate molecules will get transported into the mitochondria, whereby the completely oxidized carbon here, you can see it's oxidized, there's no hydrogens, there's no electrons really left to be used, will break off and be released from the cell as carbon dioxide. All right. Now you can imagine that carbon dioxide is basically going to diffuse from an area where it's more highly concentrated to an area where it's more lowly concentrated. It's more highly concentrated inside the mitochondria. It's going to diffuse out of the mitochondria, out of the cell, and into the bloodstream where it can be released through exhaling. So when you exhale, the carbon dioxide that's being released is actually the carbon dioxide. Some of it is the carbon dioxide that gets released from this pyruvate molecule as a waste product. So we release a carbon dioxide molecule, and also we collect, all right, we use that enzyme to put hydrogen and electrons on the oxidized form of NAD+. So we take 2 electrons and put them on NAD+ to become NADH. All right. Now, if you notice, these electrons aren't coming from pyruvate; they're coming from solution. They're actually coming from the enzyme, the coenzyme A, that catalyzes this reaction. So it's important to understand that now we're creating 2 NADH. So in this post-glycolysis pre-TCA reaction, we're losing 2 carbon dioxide molecules and we're gaining 2 NADH electron carriers. And if you're asking why 2, well, remember, if we were to go back, we create 2 pyruvates here. All right, 6 carbon, 6 carbon, 6 carbon, 6 carbon, 2 3-carbon molecules. This one becomes this. So now we have 2 3-carbon molecules going through every step. 2 3-carbon step, 2 3-carbon. We end up with 2 3-carbon pyruvates. So we've conserved the number of carbons from the beginning, 6. All right. So remember, we have 2 pyruvates here, and now we end up with 2 what is called acetyl-CoAs, or acetyl-CoAs, or whatever you want to say it. Call it acetyl-CoA. This is an acetyl group with an enzyme called CoA, coenzyme A. And a sulfur atom keeps the CoA attached to the organic molecule. So we in this middle reaction here, we have pyruvate losing in carbon dioxide. This energy is being used to reduce NAD plus to NADH, and in the presence of coenzyme A, this molecule is going to go now into the TCA cycle, or also called the citric acid cycle. It's called the TCA cycle because it's called the tricarboxylic acid cycle. It's also called the Krebs cycle by the guy who named it. Moving right along. So this step, a lot of people forget this when they're adding up ATPs, adding up NADHs, adding up carbon dioxides in the end. People forget this in the step in the middle, but it's very important. It actually takes the reactions from the cytosol and brings them into the mitochondria where our real energy is being harnessed. All right. In the TCA cycle, we're going to continue to oxidize these carbons, meaning we're going to take hydrogens from them and use them. We're going to continue to use their energy to make ATP. We're going to continue to use their energy to make NADH and another molecule as well. We're going to introduce a new electron carrier here called FADH2. It's called flavonoid adenosine dihydrogen. Don't quote me on that one. But it's another electron carrier. It actually carries electrons at a lower energy state than NADH. So we're going to make ATP, NADH, and FADH2 in the TCA cycle, which is coming up. Just important to know that FADH2 is still an electron carrier, but it actually carries electrons at a lower energy state. All right. If we move on to the TCA cycle, all right, we have our acetyl-CoA molecule. We have just released the carbon dioxide, and we have just created 2 NADHs. All right. This 2-carbon molecule binds with a 4-carbon molecule called OAA, or oxaloacetate. All right, and it becomes a molecule called citrate. It's a 6-carbon molecule. 1, 2, 3, 4, 5, 6 carbons. All right. Immediately, this molecule, and this is all happening in the mitochondrial matrix. All right, so it's happening in the mitochondrial matrix. Which is important if you want to review the parts of mitochondria. Let's go back for a second. It might help us. Oh, yeah, watching a little bit of football there, but not right now. Doing a little bio. Sunday night, 8:45. Gotta love it. I'm sure you're working too. All right, if we go to the mitochondria, we have the outer membrane, we have the inner membrane, we have this stuff in here called the mitochondrial matrix. In this blue part is really where the TCA cycle happens. Outside of it, outside the mitochondria, is where the glycolysis happens. Outer membrane, inner membrane, mitochondrial matrix. In between the outer membrane and the inner membrane is called the inner membrane space, which you'll learn about why it's important in the next part, part 4. So let's go back here and play this slide. All right. Basically what happens, citrate moves to isocitrate. I'm really going to only have you focus on some major things here. This cycle isn't as important. It's important. It's very important, and it's as important or more important than glycolysis. Glycolysis is a process that's a little bit easier to understand. There's a lot going on here in the TCA cycle. But generally what we're doing is we're rearranging carbons in a way where we can release spent carbon dioxides, meaning anytime you see a carbon with just oxygen, at some point that carbon and oxygens will be released as carbon dioxide. All right. Now, this resonant energy of the isocitrate can actually be used, and we're going to take some more electrons. All right, so we're going to take more electrons off this molecule, and we're going to put them on an NAD plus, and we're going to reduce the NAD plus into NADH. Right, so that's pretty important. If you kind of count everything up, 2, 3, 4, 5, 2, 4, 1, 2, 3, 4. We have 5 hydrogens here, and here we only have 1, 2, 3, 4. All right, so we took hydrogens with an electron, and we put them on an NADH. And we also have electrons and hydrogens from solution. Don't forget that. So the hydrogens don't actually always have to come off the molecule. In this case, only one of the hydrogens and their electron comes off. So we create an NADH. All right, and remember, everything here is being doubled because we don't have just one acetyl-CoA coming in. All right, we have 2 molecules coming in at the same time, so all of this is doubled. So we create 2 NADH here. We rearrange the molecule a bit. We lose more spent carbons. All right, another carbon dioxide here. All right, actually it's this one. We lose this carbon dioxide here. In the process, we create another NADH. Right, so now we've taken more hydrogens off, or these are coming from the acetyl-CoA actually. So CoA is an enzyme that helps catalyze reactions. In this case, it's actually binding to the substrate temporarily to carry out the reaction. So now we've created 2 NADH here, 2 more here, all right, and we've released 2 CO2s, 2 CO2s. We end up with this molecule succinyl-CoA. All right, now succinyl-CoA is basically with an inorganic phosphate addition is going to add a phosphate onto a molecule called GDP. All right, GDP is just kind of an intermediary molecule to ATP. So GDP gets a phosphate from the solution using the energy of this molecule, all right, to make GTP, or guanosine triphosphate, and in turn, the guanosine triphosphate will phosphorylate ADP to make ATP. So we get 2 more ATP here, all right. We're moving along. Succinate, all right. We have 1, 2, 3, 4 carbons with 2, all right, carboxylic acid groups here, all right, 2 COOs here. Now we're taking more electrons off, so now we have 4 hydrogens here, and if you notice in the next molecule we only have 2. So those 2 hydrogens are directly going to be added onto FAD, which is that other electron carrier, and the hydrogens are going to be carried with their electrons on this FAD molecule. So now we have 2 more. All right, we change some things around a little bit. We use the resonant energy of malate to form another molecule called NADH, which we've heard of from NAD+. So a couple of things that you need to know about this process, the TCA cycle. Number 1, it uses the products of glycolysis to occur. It needs the products of glycolysis. All right. Number 2 is the molecules get rearranged and switched around, and we take as much energy from it. And by the time the molecule comes back around, we're actually bringing 2 more carbons in. Now, where do those carbons go? All right. Well, there's 2 carbons that come in, so now we have 6, but we end up with a 4-carbon molecule. All right. So 2 carbons come into a 4-carbon molecule, making a 6-carbon molecule. Where do our carbons go? 1, 2. All right. So our carbons were lost as carbon oxides. Now the important thing to know is that the same carbons don't keep going around and around and around. There's an order at which these carbons are used. So the carbon dioxides that are left here, all right, are not going to be the same carbons that come in with acetyl-CoA every time. So these carbons actually end up getting rearranged. Some important things to know: we create 1, 2, 3 NADH per acetyl-CoA, so that's 6 NADH. We create 1 ATP per acetyl-CoA, so that's a total of 2 for 2 acetyl-CoA molecules. And we create 1 FADH2 per acetyl-CoA molecule, so we end up with 2 FADH2s. All right. So some important things to understand is how many total NADH, FADH2, and ATP are we creating in the TCA cycle, and then how many total are we producing so far. All right. If we look at ATP, how many total ATP have we produced? We produce 4 in glycolysis. All right. We're producing 2 here, so it's a total of 6 ATP. How many did we use? Well, we used 2, so we're netting 4 ATP. If we go to NADH2, we create 2 NADH in glycolysis, all right, and that's our net. we created one more per pyruvate, so that's 2 more in those pre-reactions that we just talked about. And now that's 4. And now we have 1, 2, 3 per acetyl-CoA times 2 times 6. So now we have 10 total NADH2. No, sorry, 10 total NADH, 2 total FADH2, and 4 total ATPs. All right. So now we have all this energy, 4 ATPs. Now the real energy is in the electron carriers. All right. That's the real energy. All right. We're not going to quite get to the electron transport chain yet. This is a small section. I'll give you a break on the homework. The important thing to realize here is not how to draw. Like at one point I used to have people memorize how to draw these, but not really important to do at this stage in life especially. So what the important thing to be doing is to recognize what's happening to these molecules, all right, and what's happening at each step, okay? So what's happening to these molecules at each step? I'm not going to ask you to memorize what's happening each step, but I will ask you to understand what's going on, all right? So you do need to know that carbon dioxide is lost, we create NADHs, you need to know where this happens, you need to know some of the nitty-gritty about GTP and ATP, why FADH2 is used instead of NADH. The more questions you ask in class, the more stuff you'll understand. So definitely keep writing down questions, I am a little bit disappointed that every time I ask people to write down questions, they really don't have any. But and then we get to a couple, a day or two before the test, and people have tons of questions. Please try to look over your material before you start cramming for the test. I know cramming is natural, but it's important to help you devise questions to ask in class, and other people learn from your questions as well. So that's the TCA cycle. It's the second part. It's the first part that happens in the mitochondria. It's really the primary electron producing part of the pathway. We've produced 1, 2, 3 times 2 is 6. 6 NADHs here. We've created 2 FADH2, so it's a total of 8 electron carriers with 2 ATPs. So this is really the electron harvesting part of the cellular respiration. And remember, this stuff is all driven by cellular needs. All right. So if oxygen isn't available, then the TCA cycle doesn't happen. All right. If we go back to the very beginning, all right, glycolysis gets pulled into the Krebs, and the Krebs gets pulled into the electron transport chain as a result of the presence of oxygen. If oxygen isn't present, the electron transport chain backs up, the Krebs cycle backs up, and glycolysis just occurs on its own without oxygen. All right. So let's try to keep all this stuff in perspective. All right. How do you think one might affect the other? Which one of these produces the majority of electron carriers? Well, it's the Krebs cycle. Which one produces the most ATP? Well, it's this one, electron transport chain we haven't talked about. Which one is involved with really setting up the molecule for further reactions? It's really glycolysis. All right. So great job so far. Hope you have tons of questions. I'm going to write down a few questions for you to put on a worksheet. And thanks for listening. This was the shortest part. That was part 3. Thank you.
And then CoQ. 2 H pluses are transferred from the matrix to the IM space. Electron transport chain, you make ATP with your proton gradient and coupling with ATP, same phase. Electron transport chain, you make ATP transfer with complex 2. Ooh, 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 ooh. Now CoQ moves through the membrane with its 2 E's complex 3. Electron transport chain, you make ATP with your proton gradient and coupling with ATP synthase. Electron transport chain, you make ATP. Without you, I would die. Tonight, ES hammer moves. The electrons go to the side C. 2 H's get transferred. Side C jumps over
Awesome.
to complex 4. 2 E's move to side A3. Then side A3. The 2 E's help make H2O. 2 more protons go across the membrane.
Crazy. Awesome. That brings us to part 4. Holy beep. All right. You're probably like, I can't take anymore. But you can. And you will. All right. Yeah, the electron transport chain. This is really where it all comes together. All right. Because you remember, if we were to go back, all right, we talked about glycolysis and the breakdown of glucose to make pyruvic acid or pyruvate. We have these reactions in between where pyruvate goes to acetyl-CoA, releases carbon dioxide, makes 2 NADH. We talked about the Krebs cycle or the TCA cycle or the citric acid cycle, which is really designed to make NADH and FADH2 for the electrons to use. And you probably think we really haven't created that much energy yet. I mean, we really haven't created that much energy at all. We've created a lot of electron transport molecules. We've created, what, a net 4 ATP. That's not really that much at all. And so you're probably saying to yourself, geez, like, where's all the energy? Well, the energy is coming in the electron transport chain. And this is generally the electron transport chain. It's also called oxidative phosphorylation. It occurs on the inner mitochondrial membrane. So this phospholipid bilayer here in the gray is the inner mitochondrial membrane. All right, it happens on the inner part of the membrane. This is a bad picture. It involves a series of proteins, intrinsic membrane proteins that are embedded in the membrane that actually act as electron transporters and hydrogen pumps, hydrogen ion pumps as well. It's insane. It's absolutely out of control how this thing works. I mean, so you're basically, the inner mitochondrial membrane has many electron transport chain systems. They include multiple proteins in the membrane. They're not actually showing the mobile carriers here, but you'll see them soon. They're actually proteins that move only within the lipid bilayer, which are proteins that have only hydrophobic natures. But generally, the electron transport chain is designed to pick up electrons from our electron transporters and move them through the proteins and then use their energy to pump hydrogen ions across the membrane. And the oxygen and the electrons and hydrogen ions fuse together to form water molecules. All right, and the water molecules are, generally our waste products at the end. So in cellular respiration, you end up with water molecules and carbon dioxide. So glucose plus oxygen, there's our oxygen, yields carbon dioxide, which we've already had from the pre TCA cycle and the TCA, and water, all right, plus energy, which we're going to show. So crazy thing is, how do electrons move throughout these proteins? Well, the electrons are moved from one protein to another, or within these proteins, by a series of reactions called redox reactions. Whoa! And as you can see, we have a greater electron, we have a greater gradient, we have a higher concentration of hydrogen ions out here, yet we're still pumping them that way. So this is active transport. This active transport uses electrons for the energy to pump hydrogen ions across the membrane. This is crazy. This is insane. Absolutely out of control. And so your cells are designed to make tons of energy through this process. So generally we create NADH and FADH2 in our pre TCA, our glycolysis, and TCA. And as you can see, the FADH2, which is also an electron carrier, just like NADH2, goes in a little bit down the road than NADH, just because it carries electrons at a lower energy state. All right. Now the question is, how do electrons move? They're getting pulled through. There. The electrons are getting pulled through numerous atoms of different electronegativities in order to get pulled through the electron transport chain. So another name for the electron transport chain is oxidative phosphorylation. It's phosphorylation because we're using the energy of electrons to put phosphate groups onto ADP to make ATP. We're using electrons in order to pump hydrogens across the membrane, and our ultimate electron acceptor is the oxygen here. So the oxygen accepts electrons at the end, which is important because oxygen has a very high electronegativity, and so it's going to have to be very electronegative to accept these electrons that are at a very, very, very low energy state. If we go way back, remember that electrons are falling in energy state. So the farther electrons fall, the lower energy they have, the more electronegative atoms are needed, like up here, in order to pull the electrons to a specific region or place. All right, so our electron transport chain is using electron carriers from TCA cycle and pre TCA reactions and glycolysis, drops electrons off to these proteins, pumps hydrogen ions across the membrane. And where do we get our hydrogen ions? Well, we took a bunch of hydrogen ions away from their electrons to create NADH and FADH2. So when the electrons get dropped off, we create hydrogen ions that are then used to pump across the membrane. All right. Generally, these hydrogen ions are going to want to diffuse back, but we don't let them diffuse back anywhere. We make them diffuse through this protein called ATP synthase, which is ATP to synthesize. ACE is an enzyme, so synthesize enzyme, ATP synthesize enzyme. All right. Remember, this is all happening on the inner mitochondrial membrane, which means this space in here is the mitochondrial matrix, and now here is called the inner membrane space, or the space between membranes. All right. Oxidative phosphorylation, again, it's just another name for the electron transport chain. It produces a majority of the ATP in cellular respiration, about 34 ATP. It happens on the inner mitochondrial membrane. It is generally just a series of redox reactions within proteins. It uses a hydrogen ion gradient from the movement of electrons through these proteins to create ATP through an enzyme called ATP synthase. All right. So generally, we're looking here at the phospholipid bilayer. These are intrinsic membrane proteins. These proteins, this is an extrinsic one. Its job, just like here, is to move electrons from this protein to this protein here. And here, this protein drops off electrons at cytochrome C, which is a protein, a mobile carrier as well. And it carries electrons at a very low energy state to this last one. The electrons at this energy state pump hydrogens across, and ultimately the electrons with no energy left get pulled onto oxygen with hydrogen ions to form water molecules. All right. Again, the hydrogen ion gradient that's created through the movement of electrons through these proteins. Remember, to put hydrogens across the membrane to where they are already more concentrated takes energy, right? It's basically an endothermic process. It's not a reaction itself; it's a process. But if we were to call it one, it would require energy. It would be endothermic. So we pump hydrogens across the membrane using the energy from electrons at different energy states. We allow hydrogen ions to pass through ATP synthase, which is an enzyme that makes ATP from ADP in an inorganic phosphate as hydrogen ions move through here. And the crazy thing is, let's see, that was an awesome video. I'm sure we could get a nice video of how ATP synthase works. Maybe a better one. This one looks cool. This one looks even better. All right, it's 1:26 long.
This animated sequence.
So here what we're having is hydrogen ions are going to be flowing, this is an opposite direction, but hydrogen ions are going to be flowing through the enzyme this way, and then ATP will be made here. But this is a very complex enzyme. This is ATP, very complex.
Shows the ATP synthase enzyme in operation. The animation is based on an incredible series of scientific discoveries. Only the colors show artistic license.
You can see the conformational.
ATP.
You can see the conformational change of the enzyme, and that is a result of hydrogen ions binding.
Or adenosine triphosphate is the energy currency of the cell.
And this protein enzyme is just made of amino acid polypeptides in crazy ways. And it changes configuration based on conditions resulting from hydrogen ions flowing through it.
ATP is produced by a tiny molecular rotary motor, rotating it up to 7,000
And this
RPM.
rotation is based on binding of hydrogen ions to this part of the rotor. And as they go, they bind to this part of the rotor, and then some of them go through, and that changes the conformational state and distorts the enzyme to carry out reactions.
These are so small that 100,000 would fit side by side in a millimeter.
100,000 of these fit side by side.
Protons drive the motor, unlike
Protons are hydrogen ions. They're called protons because they don't have anything but one proton. They're hydrogen ions, though, H+.
man-made electric motors which use electrons. This portion of the enzyme.
The hydrogen ions bind to this part of the motor, and then when they're done, they flow through.
Is where adenosine
and they catalyze the conformational change in structure of polypeptides to carry out a reaction that's endergonic one from ADP to ATP.
Phosphate is combined with a phosphate ion in the presence of a catalyst to produce ATP, which is then released, making way for the next cycle. A top view of the enzyme shows the sequential operation. Almost every biochemical process in your body requires ATP. Such a nanomachine exhibits all the characteristics of super
Tons
intelligent
of
design.
ATP,
ATP
tons
is
of hydrogen
vital for
ions,
life, and many of these motors were needed before the first living cell could exist. An evolutionary impossibility.
That's insane, all right? That's insane. So basically the entire reason why glycolysis, pre-TCA reactions, the TCA cycle, and the electron transport chain all exist to create a hydrogen ion gradient across the membrane by which they can flow back through to where they're less concentrated so that we can create ATP from this crazy enzyme called ATP synthase. Insane. And you're wondering how many ATP are produced in this process per glucose molecule. Per glucose molecule, we create about 34 ATP through the ATP synthase over here. 34. All right, which means 38 overall. All right. These are just different illustrations of the same thing. We got our membrane. We've got the intermembrane space. We've got the mitochondrial matrix. We've got electrons being dumped off from NADH. What they don't show here is the FADH2 part. Electrons flow through, pump hydrogens. As they do, this process of pumping hydrogens against their gradient takes energy, and the energy comes from electrons. The hydrogens have nowhere to go but back through this ATP synthase, where they create energy from that in the form of ATP, which can be used or stored in 1,000 different ways. Crazy, oh my lord. So generally, we produce, let's add all this up. All right, in glycolysis, we have 2 net ATP, produce 4 total. We use 2 in the energy investment phase. We create 4 in the energy payoff phase. So we have 2 net ATP overall, 4 total. We create 2 net NADH, and we have 2 total. We didn't use any NADH in glycolysis. In the pre-TCA reactions, we create 1 carbon dioxide molecule, but 2 per glucose, because remember, there's 2 3-carbon molecules going through here. All right. We create 1 NADH per pyruvate, but remember, there's 2 pyruvates, so we're making 2 of these. All right. For the TCA cycle, we produce 1 FADH2 per turn, but we're 2 turns because there's 2 acetyl-CoAs going through. That's 2. We create 3 NADHs per turn, 2 turns. That's 6. All right. one GTP, or let's call it ATP now, and so there's 2 turns or 2 3-carbon molecules for 2. In oxidative phosphorylation, we create water as a byproduct, and let's not forget in the TCA cycle we produce 2 lost carbon dioxides per 3-carbon molecule, so that makes 4 carbon dioxides lost. In oxidative phosphorylation, we produce water molecule, we produce 32 to 34, depending not exactly from this proton gradient. We can make about 3 ATPs per NADH, and we can make about 2 ATPs for FADH2. Let's see if you add that up, if it makes any sense total. We have 6, 7, 8, 10 NADH. So that would be 30, and then 2, 4 FADH2, plus the ATP that we've already created. So generally 34 to make 38 ATP generally. So from one glucose molecule, we create 38, or roughly 38 ATP molecules that can be used in many, many different ways for cellular work and for cellular energy. ATP will then drive most of the endergonic cellular reactions that need to occur, and will carry out many, many different mechanical cellular processes, mechanical, chemical, and transport work that goes on in the cell. So that's the overview of cellular respiration. And remember, it's driven by oxygen use. All right, so the Krebs cycle and the electron transport chain won't actually function if there isn't any oxygen available to be pulling this stuff along. Because remember, oxygen is at the end here. All right, where is it? There it is. Oxygen pulls the electrons because the electrons are being pulled by the electronegativity of the oxygen. So without this pull, the electrons have nowhere to go, and so they'll back up. The NADH will back up, and the FADH2 will back up. And if this backs up, then the TCA will get a signal not to make it, so the TCA will stop. So the processes are all basically connected to each other. The last process we're going to talk about is fermentation. And remember, fermentation, let's see. Remember, fermentation is a process that happens without oxygen. All right, so glycolysis makes pyruvate. Pyruvate goes to acetyl-CoA. Acetyl-CoA then enters the TCA cycle, makes a bunch of stuff, and goes to the electron transport chain. Remember, it's all oxygen dependent. If oxygen isn't here, then pyruvate, instead of going here, will be made into ethanol or lactate, depending on the organism. All right. Now, if you remember, glycolysis, which is the process that's primarily used in fermentation, makes a net 2 ATP. All right, and it also makes a net of 2 NADH. All right, so one of the important things about fermentation is that it occurs in the presence in the absence of oxygen. And if we start out with alcoholic fermentation, organisms that create alcohol from sugar, glucose goes through a bunch of steps, makes 2 ATP net, right? We use 2 ATP, we get 4 back, so 2 net ATP, and we make our pyruvate, 2 pyruvate molecules. We know that. We also created 2 NADH from the process of glycolysis. So in glycolysis, we know this, we created 2 ATP and 2 NADH. Because oxygen isn't there, pyruvate isn't going to go to the mitochondria because there's no oxygen present. The pyruvate then loses 2 carbon dioxides, all right? Remember, we have 2 pyruvates, so we're losing 1 carbon dioxide per pyruvate, all right? And we create this molecule called acetaldehyde, all right? Acetaldehyde then accepts, what does it do? Yeah, it takes electrons and hydrogens from NADH and gets made into something called ethanol. And this is done by ethanol dehydrogenase. Remember, dehydrogenase enzymes they either reduce or oxidize NADH. All right. So many organisms like yeast and some bacteria can produce alcohol as a result. So what do they get from this? well, they get 2 ATP. And for a lot of organisms, 2 ATP is enough per glucose molecule to keep all their cellular processes going. So, you know, the processes of fermentation of wine and beer and different types of alcohol has to do with this alcoholic fermentation of the single-celled organisms are using glucose to make ATP, but to create this product of alcohol as a result. On the other hand, we have lactic acid fermentation. Lactic acid fermentation, I think these are both the same. Oh my lord. Sorry. Lactic acid fermentation. That's not lactic acid fermentation. I can do this. I can do this. What's the best one? That's not it. What the hell is it? I want lactic acid fermentation. Yes. Please. Paste easily. Thank you. Okay, now we have lactic acid fermentation, which is going to, remember, glucose gets made into pyruvate, same thing. We create 2 ATP, great. We create 2 NADH, great. The NADH, again, is going to be used to reduce the pyruvate molecule back to something called lactate, right? So in both these processes, pyruvate, which is our product of glycolysis, gets reduced by the NADH that was created in the process itself to make a byproduct, but the important thing is that we make ATP. Same thing is going to happen here, except there's no intermediary step with the lactic acid fermentation. All right, this lactate then builds up in the cell, all right, and can make the cell very acidic if too much of it's formed. So actually, you don't actually create that much energy from fermentation. And you know, if you've ever felt that burn doing push-ups, pull-ups, sit-ups, or sprints, that you can't actually keep up this process functionally for eukaryotic multicellular organisms for too long. Our cells just use too much energy. So fermentation is a process that happens without the presence of oxygen, and is a way that cells can maximize their energy usage, but it's just generally not as efficient as the whole process of cellular respiration, which is glucose pyruvate acetyl-CoA TCA electron transport. Fermentation is just glucose to pyruvate to either ethanol here or lactic acid here, and different organisms do that stuff differently. All right. Beer, wine, bread, and cheese, all processes of fermentation are all processes of using yeasts and bacteria and sugars to break them down and create the byproducts from them. Okay. We could talk about ethanol production and renewable energy later on. I know that's a lot. That's the end of part 4. Thank you very much for listening, and I hope you're doing great. I hope this is all kind of sinking in. I hope these lectures are helpful to you, and I hope that they drive you to ask more questions in class, or at least verify some things that you're not sure about. So thanks a lot, and I'll see you in class.